Intracellular signaling components recruited to activated receptors transmit the signal onward from the membrane and amplify it. Several classes of protein are typically recruited to the activated receptors and participate in signal propagation. The enzyme phospholipase C-gamma, PLC-gamma, contains two SH2 domains through which it can bind to phosphotyrosine. It is thus recruited to the site of receptor-associated tyrosine kinase activity at the plasma membrane. PLC-gamma has a crucial role in propagating the signal onward from the membrane and in amplifying it. Phosphorylation of a tyrosine residue in PLC-gamma activates the enzyme, which then cleaves molecules of the membrane phospholipid phosphatidylinositol biphosphate, PIP2, into two parts, inositol triphosphate, IP3, and diacylglycerol, DAG. As one molecule of PLC-gamma can generate many molecules of DAG and IP3, this and similar enzymatic steps serve to amplify and sustain the signal. Production of DAG and IP3 by activated PLC-gamma is a common step in pathways from many types of receptor. Interaction of IP3 with its receptors in the endoplasmic reticulum causes the release of calcium ion into the cytosol from this intracellular storage site, immediately raising intracellular free calcium ion levels several-fold. Depletion of the endoplasmic reticulum calcium stores triggers the opening of calcium channels in the plasma membrane that let more calcium ion into the cell, thus sustaining the signal. Increased intracellular free calcium ion leads to the activation of the calcium ion binding protein calmodulin, which in turn binds to and regulates the activity of several other proteins and enzymes in the cell, transmitting the signal onward along pathways that eventually converge on the nucleus. One protein that is regulated by the calcium pathway is the nuclear factor of activated T cells, NFAT, a transcription factor. The other product of PIP2 cleavage is DAG, which remains associated with the inner surface of the plasma membrane. DAG helps to activate members of the protein kinase C family, PKC family. These are serine, threonine protein kinases that are thought to initiate several signaling pathways leading to the nucleus. Some isoforms of PKC are further activated by the calcium ion released by IP3 action. Thus, the two products of the cleavage of PIP2 reinforce each other in activating PKC as well as having their own independent effects. Many of the cellular processes that are activated in lymphocytes when antigen binds to its receptor are common to many cell types. For example, resting lymphocytes proliferate when exposed to antigen, whereas other cell types proliferate in response to particular growth factors. What differs in each case is the receptor that initiates the common response pathway in the different cell types. To link these different receptors to common intracellular signaling components, specialized adapter proteins are needed. And that's intracellular signaling components recruited to activated receptors transmit the signal onward from the membrane and amplify it. Have a great day.